welcome to the front row. My name is Jamie Williamson. Whoa. My name is Jamie Williamson, and I'm a professor here at Scripps Research, and I'll be your host and moderator today. We've got what looks like about 150 people here in the auditorium, and I think we've got well over 1,000 people tuned in virtually, so welcome to everybody in the virtual audience. As you know, Front Row is our outward-facing form of presentation of scientific research that's ongoing here at Scripps. And we feature every month a different faculty member and their research program and where it's taking new science in new directions and hopefully some new therapies. Now today, our speaker is Michael Bollong, who's an associate professor in the chemistry department. Now, the topic is regenerative medicine, and I just want to emphasize at the outset that neither Michael nor I have MD degrees, and so we're not in any kind of position to give medical research, and I'm sure many of you are interested in some detailed information about ongoing therapies. So just keep in mind, we can't provide medical advice, but there will be a time at the end of this discussion for people to ask questions. So if you're in the virtual audience, you can type into the chat or the Q&A at any time, and we will get those questions down to me at the end. If you're in person, put your hand up, and a runner will come around with a lighted sign that lets me know to call on you with a local question. And Mike and I will sit here and we'll have a little discussion. I wanted to draw your attention to some very special guests. We have members of the Legacy Circle here. These are people who have engaged in planned giving. And I want to give a special shout out to Charlie Cochran. Is Charlie here? I can't see. But Charlie Cochran is an emeritus professor. And he is the inventor of a drug called surfaxin that's used for neonatal respiratory distress, tens of thousands of patients a year. And he just, this week, is turning 94. So, I would join you clapping. I had some minor corrective surgery. That's one of the no-nos. Everything's fine. I'm perfectly okay. It was a bit of a challenge to button certain things, but other than that, I'm okay. Okay, so the subject of today is regenerative medicine. So the human body is quite remarkable. If you think about what happens from the moment of conception, you have a single-celled embryo. And that embryo contains the DNA that's the blueprint for everything in your body. And during the whole development up to an adult, there's all of these processes. And it's really this remarkable generation program. You're generating all the structures for every organ in the body. And we also know that the body sort of loses its ability to regenerate things. So many of you are aware from high school biology that you can that certain organisms like amphibians can regrow a limb if it's amputated. That's not true for people. So there's some point at which structures become, we call it terminally differentiated. They can no longer go through that process of generation to regenerate themselves. There's some remarkable parts of the body that are extremely adept at regeneration. For example, your skin, always regenerating. It's a great, wound recovery is remarkable. I'm undergoing it right now. So the liver and your blood cells are constantly being generated. So the body retains some ability to regenerate, but it's not universal. And what you're going to hear from Michael is trying to tap into those reservoirs of cells in the body that still have the potential to regenerate. And it can be for a variety of organs. You'll hear about the heart, you'll hear about lungs. But that's the essence. Can we go into the body with small molecules as drugs and tap into the regenerative capacities of the cells we have in our body? Okay, so let me say a little bit about Michael. I'm going to let him introduce himself, but Michael is first and foremost, I think I should go to, first and foremost, he's an alumnus. So he is, I think he's in the upper right corner, the tall guy in the upper right corner. And so he graduated, and he was also the first person that entered our Scripps Fellows Program. Our Scripps Fellows Program is now in about 8th or 9th year. We've hired many outstanding graduate students right out of graduate school. We track them into a proto-faculty position, and then eventually they become faculty at Scripps. It's been a terrific program. Michael was the first person to be appointed. He was the first person to be promoted to the faculty, and I think you'll see why his program is very special. Michael also has been very adept at collaborating with the Caliber Institute and using their resources to advance his programs in drug discovery. So I'm not going to say anything more about that at the moment. I'm going to welcome Michael up to the stage, and I'll join you back at the end after his lecture. So let's welcome Michael.
Terrific. Thank you so much, Jamie, and thank you, everyone, for being here. It's really, your support, your gifts, and your patronage that really kind of magnify the work that we do here at Scripps, and for that, I am really incredibly grateful, and I think many of my fellow faculty members are as well. So it's really a delight to be here to give you kind of an update as to what we've been doing over the last few years since I last gave a front row, I think in 2021 or 2022. So Jamie said I should start out by talking about my journey at Scripps. I've been here for 15 years. I started in 2009 as an undergraduate researcher in the lab of Pete Schultz. And at the time, what we were doing was looking for drug-like small molecules that could reprogram or change the identity of a normal skin cell, a fibroblast, into an induced pluripotent stem cell. And this, to me, was like this crazy watershed moment. The simple idea, or the idea that like a simple drug-like small molecule could completely change one cell type into another was really mind-blowing, and it changed my career from wanting to go into practicing medicine into actually making medicines. The stuff I saw going on here was really quite mind-blowing, and I thought that the drugs that I wanted to make, the things that I'll talk about today, really could only be made here, and that's what I think I'll focus on. So as Jamie mentioned, I did my PhD here as well. I also started as a Scripps Fellow, and I thought that if I was going to do something as crazy as starting a lab right out of grad school, I should take this as really an opportunity, like a call to arms to do something that I thought would be disruptive, right? I wanted to kind of leverage what I knew about discovering small molecules, but really wanted to turn them towards making new therapeutic drug candidates that work in the way that I'll talk about today. So that's something unique that I think you'll only really see at Scripps, and that doesn't really happen much elsewhere. Okay, so the area that Jamie mentioned, as I'll talk about today, the one that I thought would be especially impactful and something that I've been really just obsessed with since really my whole career is regenerative medicine. I think we all kind of know, like on a gut level, what regeneration is. It's this capacity that's observed throughout the animal kingdom to regenerate, obviously to regenerate, but to regrow or repair. And certainly, as Jamie mentioned, certain salamanders can regenerate their limbs, and certain organ systems, fish can regrow functional cardiac tissue. That's all well and good for the fish and salamanders of the world, right? But if I cut my limb off, I have no limb, right? If I have a heart attack, more importantly, or if you have a heart attack, 1 or 2% of the world's population dies once a year from heart failure. That doesn't grow back. So that's a burden that can potentially kill us at any time, right? So I saw this kind of as really somewhat insulting and kind of really importantly as a true medical challenge, right? We humans are often depicted as being kind of at the apex of this evolutionary hierarchy. Why can't we regenerate certain organs, right? That seems kind of silly, especially those ones that really decline as we age. So based on this, I was very interested in understanding, can we identify small molecule drugs like aspirin, Vioxx, statins, et cetera, things that would come in a pill? Can we identify something like that would endow humans with the regenerative capacities that are observed in lesser species, specifically so that we could repair in the context of disease, right? as JB mentioned, we know this is a hypothetical possibility, right? We all came from a single cell in our mother's wombs. We know that those genetic instructions are there for repair. We just need to learn how to access those instructions using the tools and the principles of chemistry. Okay, so to really start cracking the nut of regenerative medicine, we need to think about what regeneration actually is. Regeneration is making new functional cells to replace those cells that are lost in the context of disease or aging. So to do this, to make new cells, you must instruct existing cells to undergo the process of cellular division, or as frequently called in biology, and what I'll call throughout, cellular proliferation, right? So if you could do this, you could make new functional cells, you could regrow organs potentially, right? You would forestall damage, especially fibrosis, which is very commonplace in diseases of aging, and that would potentially be one way to intervene in disease. Okay, there are huge challenges associated with doing this, okay? One of these is, what are those pathways that can actually promote regeneration? What's the drug targets? What's the chemical matter that can inhibit those targets? And then, I think more importantly, how do you do that safely and selectively? Now that is a challenge that my laboratory has taken on programmatically since its inception. And we've done this quite a bit, but where we end up with our pursuits is not always clear. Anyone in drug discovery will tell you that that's the case, but how we do the process of academic drug discovery is kind of conserved across all of our projects. So in my academic chemistry lab at Scripps, we sort of start a project by asking, is there some sort of chemical matter, some drug-like small molecule that can answer some biological question that we're asking? Can we regenerate a cell type? Can we make something differentiated, et cetera? If so, we essentially bring that molecule to the stage of a prototype. Now, if that prototype is especially good, and if there's a very good reason why we should move forward, aka like a high medical need, then sometimes we've been lucky enough to work with Caliber to actually devote serious monetary resources to developing this and towards bringing it towards the clinic. For those of you who are not familiar with Caliber, it is a division of Scripps. It's a not-for-profit. It's independently funded. It has about 150 people. And the overall goal is really to put new medicines into the clinic, bring those past a point where we would otherwise take them if we were in an academic lab, right? We want to get in vivo human proof of concept. We want to show that this works in a human, and that would bring, hopefully, more monies back to Scripps and make an evergreen institute, as Pete has talked about in previous lectures. Now today, what I thought I would do is tell you a couple of success stories where we've gone down, or nearly all the way down, both the gray and the blue tracks, right? So delivering potential medicines that can go into humans, but also delivering some fundamental understanding of our own regenerative physiology. So it's in this way that I think this is kind of a cross-fertilizing way of doing discovery in general in an academic institute, which is, I think, quite unique. Okay, so I'll talk about sort of three major themes. And in the first one, I thought I would give you folks an update as to where we are with the development of a drug that regenerates the lung, the lower airway specifically, for treating various diseases of aging. So I personally have been thinking about how we might regenerate the lung for well over a decade. I have email receipts going back to 2011 talking about how we could potentially do this. It really wasn't until about 2013 or so that the science kind of caught up to figure out, to give us how to give it, what's the glimpse, how could we even do this? So we caught a glimpse about how we might do this about that time. And that mechanism for regenerating lung tissue involves adult stem cell populations. So as I'm sure many of you know, there are, in many of our organs, adult stem cell populations. They continually give rise to daughter cells, they proliferate, they make new cells to keep those organs healthy, and our lung really is no different. So there's a lot of different cell types in the lung. There's over 20, but today we'll talk about the lower airway, specifically where gases exchange. And I really only want you to remember one cell type, and that's called an AEC2 or an alveolar type 2 cell. And they're shown in green over there. And they have two functions, one of which is specialized. So they secrete surfactant. Jamie talked about surfactant. Surfactant is a soap-like substance that's secreted to the inside of your lungs so your lungs don't collapse while you're ventilating. But also these cells have a stem cell function. They give rise to these purple cells called AEC1s, and those are big squamous cells that have a lot of surface area that are required for gas exchange. So they keep they keep the lung healthy under normal periods. But why are they so important for disease? Well, they're really critically important to a disease state called idiopathic pulmonary fibrosis, or IPF for short, which I'll talk about quite a bit. Now, when you think of fibrosis, you should think about scar tissue. IPF is a absolutely terrible disease where your lungs slowly turn into scar tissue over the course of a few years. They no longer work as they should, and it really results in, I think, one of the most unfortunate ways that I think one can die, and there really are not good drugs to treat this indication. And so what we've learned in recent years is that IPF is actually a disease of stem cell failure. So the AEC2s, because of aging, mutations, and so on, this is a disease of aging, those cells no longer proliferate the way they should, and they can no longer repopulate the lower airway. So this is perceived by the lung as essentially a continual damage response, right? So in the same way, if I kept cutting my arm, I would keep getting bigger and bigger scars. The same thing happens to your lung. More and more scar tissue gets deposited. So that kind of led us to a sort of simplistic idea at the time that if you could re-endow these age type 2 cells, AEC2 cells, with the proliferative capacity that you had when you were younger, that you might be able to treat the disease at its root cause, which is something that none of the current standard of care drugs can do, not even close. So that question led us on a hunt to find a potential repurposed drug that might be able to do this. So we asked the question, is there some known drug mechanism, some known drug target that could make the cell population grow and hopefully repair damage? Now, we asked that question using the Comprehensive Drug Repurposing Library Reframe, which is a really interesting and unique asset that's unique to Scripps, as 12,000, at this point considerably more, small molecule drugs that have been in people. So every single phase 1 drug, essentially, that's been tested in a human, it's in there, plus many other compounds. So that allows us to survey a huge number of mechanisms for interesting responses. In this case, we wanted to look for making these cells grow. So what we did was what's called a high-content imaging screen. We took aged AEC2 cells from one donor, and then we asked the question, could we take many quantitative pictures treating drugs onto those cells 12,000 plus times, right? And then asking to see whether or not these cells were dividing. You can see here, this is division, this is not division. Now, at the end of the day, besides recapitulating a lot of what was known in this biology, what we found was a drug class of very widely used diabetes medications, as I'll show you, that really make these cells grow, right? So this class of medications are DPP-4 inhibitors. They're collectively called gliptins, and they're often sort of a frontline medication for type 2 diabetes. Just to give you an idea how widely these medications are used, Januvia, which I'm sure you may have heard of or know someone who is on, had nearly 9 million prescriptions in 2019. So roughly a few % of the US population are on these or have been on these at some point. So these are generally safe medications that work by inhibiting an enzyme called DPP-4. I will get into that momentarily, how these work. But the first thing that we notice is that if we take those cells and we treat them with these drugs outside of the body, either from a mouse or from a human, the effect is super striking. So multiple DPP-4 inhibitors will make these cells grow in 2 dimensions, makes them grow in 3 dimensions, and that really speaks to their capacity to make little alveoli in a tissue culture dish. It really speaks to their capacity to be stem cells. And then if we look at their gene expression patterns, it's very similar to what we observe if they're in a stem-like state. And then lastly, I show you these structures here. You don't have to memorize them, but you can see that they're all different, right? So different DPP-4 inhibitors of different structures all work. Every single one that we've tested will make these cells grow, which means that it's on target and it doesn't do something else. Now that was growth in a tissue culture dish, but what happens in an animal? Does that ex vivo expansion, that expansion in vitro, actually work to repair damage in mouse models? Now, the resounding answer to that question is yes. We've actually evaluated this drug class in 5 different lung damage models. We're damaging the lung in various different ways. inflammatory, non-inflammatory, inducing fibrosis, and so on. And what you can see here is that there's an obvious and dramatic ability to intervene in disease. So you don't have to look specifically at the details, but just to say that all of the things that look bad, such as this lung going and becoming fibrotic and inflammatory, these drugs work very well. So there's really two takeaways here. One of which is that it requires very high doses of drug, which I will describe in a sec. But secondarily is the fact that these molecules, because they are generalizably acting and providing efficacy in these models, it leads us to believe that we've tapped into something that's incredibly central in how the mammalian lung can repair itself. So we think we found something important. Okay, so you may be asking yourself, this is efficacious. How does it work? How does this type 2 diabetes medication work to expand stem cells, and why has nobody found it before? Okay, so these medications, they inhibit an enzyme. It's called DPP4. DPP4 is a degradatory enzyme, so it chews up signaling molecules. It turns them into nothing. It eats them up. It's most famous, especially in the context of diabetes, for degrading GLP-1 and GIP. These are the equivalent of your body's own Ozempic, right? Those are released whenever you eat. It results in blood sugar control and so on. So if you inhibit their degradation, you increase the levels of those guys, and they give you better glucose control. Now, what we found is that doesn't happen in the lung. It's a totally different set of things that go on. Because DPP4 is this degrader, it has many other things it can degrade besides those incretin hormones as GLP and GIP. In the lung, it's a different set. So we did some detective work and ultimately found out that IGF-1 and IL-6 are those guys that are being degraded, specifically in the context of the lung. So it's a different set of factors in the lung that are being degraded in a completely different mechanism. So some new biology that we found for what's really an old drug target. Now, if we're interested in making drugs, I said I wanted to make drugs from the beginning. Why can't we just directly go into the clinic with these molecules? Unfortunately, we've tested every single one that's gone into human beings, 13 of them so far. None of them actually have the level of exposure. Basically, not enough drug gets into the lung, the interior of the lung lumen, to inhibit DPP-4 for a persistent period of time to result in an ameliorative effect in disease. So these drugs are really meant to inhibit DPP-4 in the periphery, in your blood, your liver, muscles, and so on, not to stay in the inside of the lung. So if you see sitagliptin, for example, in the context of an oral glucose tolerance test, it's active at 1 milligram per kilogram. For lung repair, we need 100 times that. We can't go into the clinic with 100 times more sitagliptin. These drugs are quite safe, but they're not that safe, right? So that was not ideal from a direct repurposing point of view, but it did give us the opportunity to make a new drug that was ultimately much better suited to treat lung disease and that would act nowhere else. So because it's the DPP-4 in the luminal compartment of the lung, the inside of the lung, we reasoned that we could make a directly delivered drug that, because of the way it was modified, it would stick around in the lung for long periods of time, act only in the lung, and go nowhere else. That's what we wanted. And to make it safer and easier on patients, we actually wanted to dose it once per week, and we wanted to use 10x less drug than what would be required from essentially the same oral amount of drug. Now, that was an incredibly tall order, but working with the chemists and pharmacologists at Caliber, we were actually able to do that. Our strategy for making the drug is especially how it is lung retained, is shown here. So we basically leveraged several decades of known literature of molecules that have been safe and been in humans for long periods of time. We used those, and then we outfitted these molecules with groups that would give them very low permeabilities. So if we could deliver that drug to the lung, it would stick around for long periods of time. Now, we went through several hundred designs. We sent them through these flow charts of, looking for their potencies, whether they worked in animal models, and we ultimately arrived at CMR 316. This is a very potent drug. So for the pharmacology aficionados out there, EC50 is 100 picomolar, which is quite potent. And what we can see here, this is the level of the drug when we deliver it to the mouse lung. The amount of drug in the lung is a tremendous amount, and it stays in there for over a week. You can see in contrast, looking at the blood, there's a 10,000-fold difference in the level of drug. So it gets in there and it stays in there. So we think it will be safe and it will require little amounts of drug. So as you might imagine, this drug actually works incredibly beautifully in these lung damage models. In this case, we're dosing at a very low dose, 0.5 milligrams per kilogram, and we're doing it every 4 days, and we're directly delivering it to the lung. And you can see, looking at body weight, it spares body weight. You can look at the histology. This is damaged, this is undamaged. It works really pretty amazingly. And it's really, I think, kind of important to note that it's a 800-fold lower dose than amount of sitagliptin that we use to observe the same amount of efficacy. So quite a bit of dose sparing. So we think this speaks to the overall safety of a drug like this one. Now, not only does this drug work in the context of a single agent, if you co-treat with the standard of care in the same IPF model, it also shows efficacy, but it actually synergizes with the standard of care drug. So that is an effect which is greater than additive. So it means those two things working together give you a very, very good response. So nintedanib is really kind of a repurposed cancer drug that only sort of slows down the scar tissue deposition phase of the disease. It doesn't do anything else. So adding on that regenerative component to this model really gives you an efficacious effect that's greater than additive. And this actually helped us design our clinical trial in the sense that we wanted to be able to both dose this agent alone, but we also wanted to see if it would improve the efficacy in patients, the overall outcome, when co-dosed with the standard of care, whether that be nintedanib or another drug that acts very similarly. So luckily, we think this drug will be quite well tolerated because the drug is quite potent. Our calculations tell us that we'll only need to dose 2 milligrams of the drug once a week in the clinic, which we think hopefully will be well tolerated. So as you can imagine, we are, based on this data, quite excited about moving forward into the clinic with this molecule, seeing what it can do in patients. This project started around 2017 when I started my independent lab, as Jamie mentioned. And in 2021, we were actually able, with Caliber's help, to declare the clinical candidate, CMR316, that I just talked about. That's about the time that we had the last front row lecture. In the last few years, we've completed the very expensive, arduous, long-lasting GLP toxicity studies on this compound, and we actually submitted an investigational new drug application to the European Medicines Agency, it's called a CTA, to allow us to be able to dose this in human patients. So it's actually pretty amazing today to be able to announce to you all that as of next month, we should be dosing in human patients at the Fraunhofer Institute in Germany. It's a specialized lung institute evaluating both the safety, the PK, so like how much drug goes where, and the target engagement in healthy volunteers and a couple of IPF patients. So if all goes well, at the end of next year, we should know whether this is well tolerated or not. So it's quite gratifying to see that this has already gotten to this stage, and that's something I think could only be attested to because of Caliber. All right, so before moving on, I thought I'd kind of recap a few points about this project, namely that this is really the only regenerative approach towards treating IPF. It's the most advanced among these. Pharma is quickly catching on to the fact that this is the root cause of the disease, and they're scrambling to get drug molecules like this. But most of their pipelines are still filled with drugs that work, like nintedanib. They're anti-inflammatories, they're anti-fibrotics. Now, also, we think that these mechanisms will likely be safe because IPF really is a disease of inefficient or ineffective amounts of proliferation. So restoring these back to baseline really only uses the body's natural mechanism to bring it back to a normal state. And then lastly, I do think that especially this molecule, will be applicable to other potential lung diseases, right? Everyone asks me about COVID-19. I think the answer to that question is probably a yes, but we've been focused on probably our next sights are set on COPD. This is a really tremendously unmet medical need. It kills 5% of the world's population every year. So if we can have an impact there, I think we would really be quite happy. And just in general, we're quite optimistic about what we can do with these molecules. Hopefully it's that we can treat degenerative diseases, and hopefully we should know soon what's going on in the clinic. Now, as I mentioned, this was an absolutely massive effort. It took a lot of people besides myself to get this across the goal line. As I think multiple people have said in these front row lectures, drug discovery really is like the ultimate team sport, and it's a great example of if you want to go far, you gotta go together. And so at this point, I'd like to sort of acknowledge some of the key folks. Sita did essentially all of the in vitro and in vivo work. Pete was a champion of the project since the beginning. Sean led it across the later stages of development, and Jeff was one of the many chemists who worked on the project, and he designed the molecule. And then lastly, before I move on, I'd like to say that this drug development campaign, especially one particular part, the IND-enabling toxicity work, the GLP tox work, was actually founded by Ed Bachrach's Family Foundation. So I think it goes to show that no matter how much you can give, right, whether it's little or quite a lot, like Ed's foundation, if it can really go to funding tangible translational research that can potentially make a meaningful impact in patients' lives. Okay, so that was a drug discovery campaign starting from what we'd call a phenotypic screen, looking for a very specific purpose with no particular target in mind. But we've also been focusing on drugging known regenerative pathways in mammals. Now, as I showed you from the first few slides, as you might guess, there's probably not a lot of known regenerative pathways in mammals, and that's true. But one that is well known is how our organs, at least in part, control their size, and that's a pathway called the Hippo-Yap pathway. So you can think about the Hippo-Yap pathway as basically two components working in opposition. YAP is this gas pedal telling the nucleus that the cells need to divide. They want to divide until they touch each other, until organs get to a certain density, a number of cells touch each other, and then the Hippo pathway is activated, and this results in the inactivation of YAP. So it's this interplay between these two factors, which in some ways controls how big our organs get. Now, this is actually a very well-conserved pathway. It was discovered in flies about 30 years ago, and you can see that if you start knocking out the brakes of this, right, the Hippo pathway, you get very large organs. So for example, you get large heads and wings and all sorts of malformations in flies, and if you knock it out in the mouse, you get very large livers, and some organs will also get large too. Now the key question, I guess, that you're asking is, why does organ size control have anything to do with regeneration? Well, it turns out that YAP is actually being cycled on and off all the time in the vast majority of our organs just to replenish them under normal circumstances, right? So when we are in utero, actually YAP is on at super high levels. However, once we're born, it gets turned on only cyclically, especially in the context of disease or damage, so that you can repair and go back to sort of homeostasis. Now, the problem becomes that in certain disease states, there's an insufficient or no YAP response at all, and that results in ineffective proliferation and no repair. So that led us, again, simple-minded idea, right? Not enough cells make cells grow. Can we turn YAP on when it was supposed to be turned off normally? And would that result in regenerative repair of a given organ in question? Now, that really sort of simplistic idea has led us on a very extensive campaign that we've done several times to really interrogate these large chemical libraries for molecules which might turn YAP on when the Hippo pathway was activated. So the way that we've done this is screen very large libraries, on the order of more than 700,000 molecules. We find small molecules that turn this transcriptional pathway on. We then figure out how those drug targets work in cells. That could be its own lecture on its own. And then ultimately, we use cell biology to figure out how those molecules work in cells, and then in some rare cases we take these into animal proof of concept studies. And just as an example, something that I don't have time to talk about today, but we've shown that sort of in an in vivo setting and an ex vivo setting, we can regenerate skin. We can make old human skin look like young skin, which is really something phenomenal that I'd be happy to talk about. And we can actually heal wounds faster in pigs as well. So we've been lucky enough to turn the crank on this sort of discovery engine a few times, and it's been, I think, incredibly productive exercise for my lab, both from the perspective of developing new drug targets, but also just learning just essential basic science. So every time we figure out a new drug mechanism from scratch, we actually end up annotating new components of the pathway, which are things that people have not found by doing classical genetics. So in this way, we're really both finding new drug targets, but using pharmacology as a way of doing genetics, which is something that I ultimately wanted to do with the lab as well. So I won't go through all of these other than to say that we've done this exercise now a lot of times and come up with about 15 different mechanisms, 15 drug targets for doing this. And I think that's something that's unique to my laboratory and to Scripps. So now that we've got this huge palette of potential prototypes of YAP activating drugs, what can we do with them? What disease should we go after? And I said, why not just go after the biggest unmet medical need there is, and that's It's heart failure, right? That's still the #1 killer of men and women, both in the United States and worldwide. Cancer's catching up, but it's still really quite terrible. Roughly a few percent of the US population has heart failure. It's a $30 billion healthcare burden on our system every year. And we've made, I think, great strides in sort of treating and in some ways preventing the patient's decline, but there really are not drugs that repair the damage that occurs to the heart, right? There's no way of regenerating the heart, like I showed you in the beginning with that fish. So the most common precipitating event, the thing that causes heart failure, are heart attacks or myocardial infarctions, as I'll call them, MIs, right? The cardiomyocytes, the muscle cells of the heart, they die and they don't grow back. This causes irreparable damage. You get scar tissue, you get maladaptive remodeling, death, right? The thing is that these cells do not grow. Like the same cardiomyocytes you have when you are a teenager are roughly the same ones that you have when you're 75 years old. So this is a non-regenerative organ. So that led us to the very simplistic idea that we might be able to tell cardiomyocytes to regrow in the context of damage and hopefully repair the damage of the infarct with a small molecule that activates YAP. This would effectively repair damage and in principle forestall a progression to heart failure and ultimately maybe a cure, right? So that was a just pie in the sky goal that we had been thinking about for a long time, but really within the last few years, Jim Martin and a number of other just pure genetics biology groups have shown that this is an actual possibility. So they've used genetic engineering in both mice and pigs to show that if you inhibit Hippo, so essentially if you activate YAP, you can repair damage, you can reverse the damage that occurs after a heart attack, right? So they're surgically inducing MIs, and for example, these rodents, you can see these guys have much less muscle. You inactivate Hippo, you have a lot more muscle, and you regain cardiac function. This is 3 weeks after a heart attack. Same thing is true for pigs. Now, this is truly phenomenal. I get excited looking at this every time I do, but genetic engineering in human beings is still a ways away. It's still very much in its infancy. So we thought a much more expedient and ultimately many times more scalable way of doing this would be with a small molecule drug that could turn YAP on locally in the heart and repair damage. So just kind of skipping several years and pain of trying to discover these guys, I thought I'd tell you just about our clinical path, right? So we're making small molecule drugs. These are often delivered, right, as pills orally, right? But one of the key things about my program is that these drugs must be delivered locally so that they act only at the site where we want them to and nowhere else. So we thought we would deliver this locally to the heart. There's been advances in cardiac delivery, so the anticipation and what we're working with now is that we can deliver these to a sac that surrounds the heart called the intrapericardial space. The key goal ultimately would be that we kind of deliver a cardiac patch in that intrapericardial space, and the drug would slowly bathe the infarct over the course of a week and repair damage. And that's essentially what we've done to date. Now, our envisioned therapy, as I show you here, is we think about it as the patient has an MI. Before they go home, a few days later, essentially the day before potentially, they would go into the cath lab for a one-time injection of drug. It would take potentially less than 30 minutes. It's a non-invasive procedure. And the anticipation is that ultimately this would be done by an interventional cardiologist. Potentially thousands of people in the United States could do this and wouldn't require a cardiac surgeon. So it would be, in principle, a routine procedure that you would get if you had an infarct of a certain size. Now that would be a one-time therapy that would hopefully save you from progressive decline and cardiac death. Now, we've been lucky enough after several years of trying to test this hypothesis in a mouse model of MI. So we, like the data I showed you from Jim Martin, we can surgically induce a heart attack in the mouse. We can give them MIs. This is the only data that I'll need you to interpret here, is that we look at left ventricle ejection fraction. So that's how much of the volume of the left ventricle pumps every time it pumps, right? So normally it's 70% of that volume is expelled. We give them mice MIs and we give them just absolutely brutal heart failure, right? 20% decrease is a pretty bad, but now we're giving them 40% decline. And what we see is when we dose one time with one of our drug prototypes, it goes back, and that's 7 days. So we've regrown a lot of that potential capacity very quickly. the disease becomes worse as the model goes on, and you can see that we're still having an effect here. And the effect is functional if you look at the M-phase echocardiography. So this was our prototype. This is kind of the data that we're circulating, but I can tell you now, for the purposes of today, that our actual clinical candidate actually goes almost back to normal. It's in the 60s and 70s, which is just truly amazing. So we've been advancing molecules like this one into later stage models. We've shown that they actually expand cardiomyocytes in the pig. Mice and human hearts are pretty different, but human and pig hearts are pretty similar, roughly the same size, roughly the same physiology. So this is really the gold standard in being able to evaluate these agents. And so what we've shown is that we observe a very long-lasting effect over the course of a month from a single one-time injection of one of these drug prototypes. And we don't have a farm at Scripps, so we have had to rely upon a couple of our collaborators who are really quite experienced, both in the delivery and in doing these models in pigs. So we're really excited that this will have an effect in human patients, hopefully in the near future. And if we're successful in the later stage preclinical development of this program, of which we're doing right now, we're anticipating being able to submit the IND towards the end of next year with patients probably around 2026. So again, I'd kind of like to linger on this just briefly. You know, there's been a lot that people have put out there, but to our knowledge, this is the only pathway. YAP is the only pathway that will make cardiomyocytes expand in an adult organism and not induce arrhythmias. There have been people who have put reprogrammed stem cells in. They make the heart do this. You don't want that. This is a program that's turned on when you are in utero, right? Your heart is both growing and it's increasing in cell number, right? So this is what happens when you're essentially a baby. So we're recapitulating that in an adult. I think this is the first pharmacological agent, so the first small molecule that's gotten this far. And then lastly, again, I think this will be broadly applicable. If we can show good effects in the post-MI setting, so people who've had heart attacks of a certain size, I think this will have potential broad applicability to others. Even potentially, you know, this is just a hypothetical, like since in lots of people, the ejection fraction goes down over time. Certain people stay the same, but certain people, you may just get a tune-up every so often and you could potentially repair your heart, or at least that's the envisioned future. Okay, so in the last few minutes, I thought I would talk about something that's newer, some earlier work that we're quite excited about. And this involves drugging what I'm calling the cellular causes of aging. Now, as soon as you mention aging research, you get into kind of a tricky area, but I thought that if we stuck to biological pathways that were really well conserved, really well characterized by decades of data, interventions that have been proven by many people, we may be able to bring some expertise as drug developers, as chemists, as chemical biologists, hopefully to develop tools that could modulate known longevity phenotypes in mammals, which would be something I think that's worth pursuing. Okay, now I thought I'd talk about two of these. The first of them involves something that happens as we age, and that is our DNA loses its integrity. So every time you undergo a cellular division, the ends of your chromosomes, the ends of your DNA, they degrade just a little bit. Now there's an enzyme that adds back those missing sequences, that missing bit of nucleotide, and that's called telomerase. Some of you may have heard of. But the problem is that telomerase is really not expressed in the vast majority of human tissues. It's in some stem cells, but essentially nowhere else. Now, our collaborator, Rhonda Pinho, has showed that if you turn telomerase on in an old animal, that you get a bunch of very interesting anti-aging phenotypes. Like their brains get bigger, they have better memories, better resilience to certain neurodegenerative phenotypes, and so on. So that led us on a hunt with Pete Schultz and Rhonda Pinho to try and find a molecule that could turn telomerase on in both mouse and human tissues. Now, we identified several molecules, one of which we're showing here. We very cleverly called it telomerase activating compound. Now, what it lacks in its name, it really makes up for, I think, in its activities. So it has a slew of really interesting phenotypes that happen in aged animals. You get increased neurogenesis, there's decreased inflammation that's associated with aging, and there's actually a loss of senescent cells in a number of different tissues like kidney and spleen and so on. So this is very much a prototype. maybe even a prototype, right? This molecule is super simple. It's like one step away from being as simple as aspirin, for example, right? We don't know exactly how it works. We don't know the drug target, but I think it would be especially interesting to understand this target, maybe even make a drug that acts like this, to potentially intervene in diseases of aging in the future. And Ron and folks like them will bring these forward, hopefully with our help. Now, in addition to some of these sort of anti-aging pathways, I thought we would like to also sort of recapitulate some known longevity interventions, right? One of those is caloric restriction, and I'm sure that you've heard about this. This gets quite a lot of playtime in the news and so on. If you feed lots of, essentially every mammal, this has been tested in a well-controlled trial. You get mice, dogs, monkeys, 30% fewer calories over their lifetime. They live considerably longer and healthier lives. We, of course, wanted to find drugs that could do this, right? Because if you could do that, that would be tremendous, right? But we wanted to be able to do this without having to starve the people that were taking them, right? So what we do know is that this phenotype, this caloric restriction phenotype, is almost exclusively mediated by a pathway called mTORC1, which is quite famous as well. The problem is that there are not good mTORC1 inhibitors that are full or selective. There are some that are selective but not full, and there are some that are full but not selective. So if we could do this, it would be something that's really, really tremendous. So we're on a really massive campaign to try and find. We're evaluating over 10 billion molecules for a compound that may inhibit this pathway. If you could do this, this type of drug would be absolutely amazing. You would lose weight, you'd potentially live longer, you'd have better glucose control, and based on the literature, you'd have better memory too. So we're still at the very beginning of this quest. I'm cautiously optimistic that something may prove useful in this, but we're excited to see where we'll end up. Now, last slide. I thought I would leave with kind of a forward-thinking goal about what we want to do, right? So I talked about drugs that would regenerate the heart, regenerate the lungs, but what about all of our other internal organs, right? There are a lot of other diseases besides the one I mentioned today. And my goal with the lab, hopefully by the time my career is over, which I don't think, or I don't hope will be soon, but is to go in and ultimately develop a drug that will act to regenerate every internal organ and bring it, hopefully, to the human proof of concept stage. Now, we're at this. We're aggressively working at this. For example, we've got molecules that will regenerate parts of the retina, the cornea. We've got molecules, or we're looking for molecules that will regenerate new muscle. We've got compounds that protect the GI for IBD, and the list goes on. I think sometimes to the consternation of my trainees, but we're giving it our best shot. Now, ultimately, our goal is to, as I mentioned before, live longer and hopefully much healthier lives by really tapping into a regenerative potential that I think is within each of us. So I will leave the talk at that. I'd first and foremost like to thank the folks in my lab who obviously did all of the key work. They were the ones who kind of believed in the dream of actually doing this and actually did everything. So I'm quite grateful for them. As I mentioned, yeah. They're right there, by the way. They're in the front. I mentioned Sita did most of the work for AEC2. Edita was really the person who started a lot of our work in YAP. Kayla and Maya have been working a lot in that space as well. Thanks to Kayla for a lot of the artwork that has been in this slide deck, especially this particular background, which was hers as well. I've also had a lot of great Caliber and Scripps collaborators, and we've been grateful to receive money to both fund our academic research as well as a lot of drug discovery that we've been doing. So thank you very much again for your kind attention. I'd be delighted for any questions in the Q&A session. Thanks.
Okay, thanks, Mike. Now, just to remind everybody who's here, you put your hand up and we have a runner that will have a little lightsaber that we can that locate on you. So, Mike, let me just start out while everybody's organizing and ask a question. So you described YAP as the gas pedal.
Yeah.
And then Hippo was the brakes. And so you clearly showed that the YAP targeting was good for proliferating. But how does that translate to the rest of the systems in the body? I mean, is YAP in every tissue? Are you firing everybody up? And how do you kind of zero in on what you need?
As you can imagine, it's incredibly complicated. So Hippo is kind of a nebulous thing. It's different in every cell. It's different in every organ. And what we found is some of the mechanisms for turning YAP on, inhibiting Hippo, they don't work in certain systems, right? So some of the molecules that will expand cardiomyocytes, they have no effect in the liver. So it's very different depending on where you are. And so we're ultimately, what our goal is to play sort of mix and match. What's the best drug target, best way of inhibiting Hippo for a given indication? So that's critically important.
Good. Yeah, we have one over here. That's a question from the audience.
Hello. I'm sorry. That was really interesting. Thank you very much. I two questions, if that's right. Firstly, the drugs that you're looking at that are promoting cell proliferation, do you see any increase in things like cancer as a result of those drugs? And secondly, there was a news story today from a group in the UK that had looked at IL-11 suppression. I wondered if you had any comments on that as well.
I'll tackle the first one first. I think that's a great paper. It speaks to the importance of mTOR specifically. So I think if you can do that in every tissue, not just the ones that are affected by IL-11, that would be tremendous. So I think that's great. And I also read that and I'm excited by it. To speak to the first question, yes, it's certainly a concern always that we will observe overproliferation. Now, it's been our experience that we've never observed any so far. So we've taken a lot of different steps to mitigate that possibility, right? So because of time and just the audience, I usually go through a slide that details our approach, right? So we don't get off-organ proliferation because our drugs will always be targeted, right? They're intrinsically fast clearance, so as soon as there's dissolution from the organ in question, they're very quickly cleared, so there's not enough drug to act anywhere else, so it acts really only there. And then most often, we're going after disease indications where we don't have to intervene for long periods of time, ideally not chronically. In the context of, say, the heart, which I talked about quite a bit, we think that will be a one or several time intervention, and the likelihood that you have a cardiac tumor is incredibly low. There really aren't any. I mean, there are, but they're very, 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 very rare. So in those cases, we don't think that we'll observe overproliferation. And just to add a little bit on that, we've also just gone into gross tissue, like we've applied it directly to the skin. We see proliferation to a certain point. It usually doesn't go past a certain point. And what's interesting is that we don't just reset all of these tissues, they go back to normal, which is surprising. So if you take the drug away, it goes back to normal, which is interesting. So there's a lot left to understand, but ultimately, right, the ultimate proving ground both for efficacy and for safety, like cancer, is the clinic, and we need to evaluate this in patients, and that's something beyond what I'm doing.
Yep, it's terrific, thanks. So a question from the virtual audience. So I would note that you became increasingly ambitious throughout your talk in terms of number of molecules you were screening. And it started out with 10,000.
I wanted to.
Then you got to 700,000.
Yeah.
And now you're up to, you were in the billions, okay?
Yeah.
So the questioner was asking, how do you screen so many molecules? And I think, it's really speaks to the power of what you can do at Caliber. But maybe elaborate on that.
Of course, I'll just start out by saying Caliber is incredibly impressive. There's lots of different ways of doing these types of screens. I'll just say that those types of screens, they're scaled and they have different purposes, right? So in the beginning, we started out with a screen that has a high level of information, an imaging screen, so we can't screen 1 billion molecules, right? So we screen the ones that are most valuable. To screen 700,000, we have robots, and we can screen them using cells and look for turning on a gene, for example. In the billions, you have to do a DNA-encoded library screen, and I'm sure that folks have talked about this in this lecture series. So you have to know what the target is you're going after and then do it in a vitro setting. So I think in each case, it's just a matter of using the right technology with what you're going after.
And another question that came from the virtual audience was, are you able to use AI in any stage of this to try to improve efficiency? What's going on there?
Yeah. On the discovery side, It's been less useful for people discovering the basis for new drugs. I think that there's a huge potential for AI, but I don't know if it's at the stage of discovery yet. I think it's been incredibly powerful in saying, once you have, say, a compound and you know its target, can you make a ligand that's better suited towards that? That's, I think, tremendous, and people have shown that many times over. It will be the new proving ground of, can you take all of this information, and folks like Arnab, who's in the audience, are working on things like this at Caliber, which is to say, can you mine huge infrastructures of like, say, literature and patents, and then combine them and say, can we find things just de novo from this information? I think that's the next thing, and I'm excited to see where it goes.
Well, as a little test, during your lecture, I typed into ChatGPT, can I have a drug that regenerates my heart tissue? And it said I should do stem cell therapy. So with that, but there's a serious question there. So there's, many people have heard about stem cell therapy, where you're programming stem cells and then reintroducing them into the body to try to do some of these tasks.
Yeah.
And that's in distinction to the approach that you've talked about today, which is using molecules to activate the stem cells we already have. So give us the over/under and comparison, and
Yeah.
it's okay to pontificate. It's okay.
Yeah. So obviously stem cells are tremendously important. I think manipulating them specifically in the context of the endogenous stem cells, the ones that are in our body, is probably the most powerful way to do this. We just need to find the right drug targets and drugs, as I mentioned. Stem cells have been used a lot, and when you say stem cells, it can mean many different things, right? Some people go to clinics in foreign countries and they inject their joints with stem cells, and some people have a great time with that and they observe efficacy. Some people put reprogrammed cells that they made in a dish into hearts. That's a different thing. And people have done both, actually, in hearts. Now, the problem with those, especially in the context of the heart, is those have been just, unfortunately, just an abject failure. They have not really provided long-lasting efficacy in the clinic. It's a tough area to go into, and we're well aware of that, but I think it's worth it. So stem cells can be powerful, and I think that there's great applications potentially in the future for them to take an exogenous stem cell and put it into our body. For example, people are making like little pancreases that you can put into yourself to intervene in type 1 diabetes. I think that's a great use of a stem cell. I think you just have to be thoughtful. It was worth the stuff that we did, not we particular, but collectively we in cardiac. But I think we're taking a different approach. So.
Right, and you're clearly getting some traction.
Hopefully so.
Do I have any other local questions here? There's one. Yep.
Thank you so much. This is absolutely a brilliant introduction to some of us who are not in this industry, but I am in clinical trials industry. So I have a question. So you do have a division where you're taking all this brilliant research to clinical trials. First question is, please tell me a little bit about that. Second question is, how are you promoting these ideas, this brilliance, to the biotech companies and big pharmaceutical companies? What are your collaboration, aspirations with those companies? Because they can really take you forward.
Absolutely. So I'll first start out by saying I am the earliest person. I'm the person who comes up with the idea and gets it frequently to the prototype stage, and then working with the other brilliant folks at Caliber, we can take that to a much later stage, to IND stage, and then, in some rare cases, actually becoming less and less rare, right? There's 10 INDs at Caliber within roughly 12 years, which is really phenomenal, right? So Caliber has their own late-stage preclinical team. They have their own clinical team. They have a CMO who's designing the clinical trials, and then we work with CROs to actually execute those trials or do investigator-led clinical trials, depending on what exactly it is we're doing. So they've really massively expanded their infrastructure for doing clinical trials and establishing, hopefully, proof of concept efficacy, or at least safety.
Okay, Michael, we're coming up on the hour, so maybe I'll ask the last question, which is, you told us all the great stuff that's got a lot of hot results. So what's the next big thing? What are you working on that
Yeah.
you're going to tell us about in 2 years that you're excited about?
I'm excited about it all, otherwise I wouldn't do it. But the one thing that I think we're gaining a lot of traction with quite quickly is another really tremendously large unmet medical need, and that's in the context of dry AMD, so macular degeneration. It's the most common cause of blindness, I think, at all, and it's very common, especially as you age. I think my grandmother actually has it. And these people, they can't see their grandchildren, they can't watch TV, they can't read a book. It's absolutely terrible. So what happens in that disease is that there's actually loss of the back of your eye, the retina, and as that happens, you kind of have an epithelial layer that dies, then your photoreceptors die, you get drusen and all of this. So we've got molecules that will expand those epithelial cells, hopefully so that we can reverse or at least stall that loss of photoreceptor cells. So I think if we can do that, especially from a one-time injection, sort of like the heart, that would be tremendous, and that's, I think, one of the largest opportunities that's unmet in general in the ocular space.
I'm sure there's others, but we'll look forward to having you
opinions.
back to talk about this. So, and just in conclusion, we have next month we have Front Row, and it's Arnab Chatterjee, who's at lead scientist at Caliber, and that's, I believe, August 21st. And so we welcome you to tune in again virtually. We encourage you to come to campus and sit here. For those of you who are here, we have a little reception as an incentive. And we look forward to seeing everybody next month. And when you're here, you will see me in the front row.